Again, thank you everyone for joining. We are really excited to have you all here. We really hope you will enjoy these 2 days, and we really hope you actually find it really beneficial, and hopefully it will be helpful for your research. With this, I would like to introduce our next speaker, who is Dr. Tim Foley. Tim is the lab head of DNA-Encoded Libraries selection and pharmacology, and a senior principal scientist at Pfizer. Tim is also the head of the organizing committee of this workshop, and will present to you today an introduction and an overview of this workshop. First, thank you so much, Tim, for all your efforts organizing this workshop. We're very thankful for you, and welcome, and the floor is yours.
Hey, well, thank you for the kind introduction, Serine. I'm checking very briefly to make sure you all can see my screen and hear me.
We can see you, but not, okay, we can't see what you're sharing, Tim.
Okay, let me try that again. Share.
Yes, we can see what you're sharing. It's your desktop.
Yes, there we go.
Wonderful.
Great. All right. Well, thank you, Serine. Now I have a window on the right-hand side of my screen, so this is great. So thank you for the kind introduction. I want to cover briefly the who, what, why, and how of this event that you have just signed on for. So just wanted to start briefly with an introduction to DNA-encoded libraries, and specifically why I think it is making such a big splash and there's so much attention around it. And I'll start by saying first that many of us on this call are pharmaceutical and biomedical researchers. And we're all focused on the goal of trying to find new therapies to address unmet medical needs for patients. Now, many of those therapies that we seek to develop will affect their action and improve patients' lives through the use of a small molecule therapeutic drug. And in order to develop this end goal of a small molecule modulator that impacts patients' lives, we need a starting point. That starting point is accomplished through hit discovery activities at the very early stages of a project. Now, I have shown here in this slide a table that summarizes key attributes of the different ways that we go about finding these hits and chemical starting points for our projects. Now, I won't belabor the details of this slide, but to say we've got 4 major ways that we do this in the industry. The first 2 are through either diversity screening or virtual screening, where we look back and utilize our corporate chemical collections or commercial collections that we purchased and develop screening assays, as Anton described, and look for compounds that modulate the target or biological function that's being interrogated in the assay. Now, these both rely on our corporate and chemical collections. And another emerging platform for doing hit discovery for target-based programs is affinity selection mass spectrometry. This uses the same principles of the same biochemical principles of hit discovery that are applied in DNA-encoded libraries, but uses small molecule mass spectrometry to determine whether or not the molecule is bound rather than a DNA barcode. Suffice to say, this small molecule ASMS technique still relies on our corporate chemical collection. So when we think about which of these are we going to use to identify hits for a project, we were really excited by the idea of DNA-encoded libraries, because no matter which of these first 3 that we select, we're all using the same compounds that are there already in our holdings. And this collection can be biased by the therapeutic areas of focus of your organization. And if there's a change into new areas or modalities or target classes, you might be the chemical matter that overlaps and acts on your targets may not be present in that collection that you screen. So we were really excited. And I think the excitement of one of the big excitements of DNA-encoded libraries is the idea of accessing these very large and chemically diverse collections of unprecedented size, ranging somewhere. We count DNA-encoded libraries in the billions of compounds, and this is 3 to 5 orders of magnitude larger than the chemical collections that are held by large pharmaceutical research areas. So these are very large chemical collections that are unbiased by what we've already done, so that we have the opportunity to find things that are new. Overall, it uses very low protein compared to the other screening platforms, which means we can get into this and identify hits for new targets or proteins that are targets that are difficult to purify, and which is a shortcoming of other discovery campaigns. So we don't need much biological material to get into here. When we do the screening, it creates a rich biological profile. We can generally run the DEL screen under multiple conditions and allows us to pick the hits with desired mechanisms of action that we may suspect have a better chance of translating for patients. We find that generally it has a broad applicability to target-based drug discovery programs, and in the pharmaceutical sector, this covers about 50% of our portfolio. We've seen some interesting and emerging technologies to use this on whole cells and access membrane to proteins and other classes of targets that have not been previously interrogated with the technology. So I'd say this is an area of growing capacity and with the technology in the field, but to say that it's sufficiently large that it's applicable to the entire sector. As a hit discovery scientist, initially trained in high throughput screening and classical pharmacology, I really appreciate that DEL has a very low hit rate. You can be frustrated by the fact that you screen billions of compounds and only get tens of things to follow, may only get tens of things to follow up on at the end of the day, or sometimes nothing. But we find that in general, this low hit rate allows us to prioritize what we're working on and focus on things to follow up that have a good probability of being hits. And you'll hear a lot in our session tomorrow about how different people are approaching confirming the hits that come out of DNA-encoded libraries. We also like that it's fast relative to a full deck HTS, and that it has a low cost of use and per project basis once a platform is established. So there's really a minimal investment that's required to make proteins and screen targets once we have our DNA-encoded libraries built. So I want to transition, and so those are DNA-encoded libraries, why we're interested in them. I want to start by thanking the organizing committee. It's shown here on the slide: Abigail, Anton, David, Gwen, Canny, Kyle, Lisa, Matt, and Serine. And also a special thanks to Mike Marshali. He is our technical wizard behind the scenes making this virtual event possible. And a special shout out to Abigail Grossman, who drove the application and implementation of this webinar. So, and the entire registration and operations of this workshop. So thank you, everybody involved. I think we put together a really fantastic program for you. And to say the goals of this workshop are to share fundamentals and applications of DEL technology. And I hope that we share details about that with you that includes the tribal knowledge of how to effectively use DELs to discover hits. This is a particular focus of the Assay Guidance Manual, and how I found the AGM in my graduate studies back in 2007, trying to understand how to build an assay, design it, and get it ready to run a high throughput screen and pharmacologically characterize compounds. The information in the AGM that is just not out there in any other textbook, hoping that we embrace the spirit of that in the presentations that you'll see over the next 2 days. Hope to have candid discussions about the pros and cons of the various approaches to using DEL, so that the people who are on the call and in attendance can have a deeper appreciation of when DEL might work for them and when the risk for challenges or failures becomes high. I'm hoping that we can engage in a dialogue about the technological hurdles that exist to the advancement of the field. And we will have a particularly interesting session in the end of tomorrow that will cover emerging trends at the forefront of the field. And I think we'll see a clear identification of the technological hurdles to seeing those advances move forward. The objective here is for novice users to take on practical learnings that can be immediately applied to research and improve the outcomes of your work with DNA-encoded libraries. And I'm hoping that experienced practitioners, those of us on the call who have been working in the field a while, or that we develop a deeper appreciation through better understanding the complexities of the areas that are outside our original expertise. So what is this workshop? With the organizing committee, we pulled together 4 key sessions that really follow the workflow of DNA-encoded libraries and share the information with you, so that we hope that builds your knowledge and appreciation for how all of these aspects of DEL operation are interconnected and dependent on each other. So we'll start, or I'll start by noting the 2 plenary events of the session. Dario Neri's keynote talk coming here shortly, as soon as I'm done. And then, as Anton mentioned, Barry Morgan's Great Moments in DEL History, hosted at the end of the day today. She will certainly be an educational and interesting event for all of us. I'll just say thank you, Barry, for pulling all of that together. I'm really excited for this. So the 4 sessions we've developed include start with DEL chemistry, because for any hit discovery, you need a library to work with first before you can start thinking about screening it. So we'll hear talks from Chris Hupp and Yun Ding concerning how to design, thinking about how to design DEL libraries, and then how to actually apply the design and the chemistry that's out there to manufacture a DEL library so that you could consider screening it. In session 2, we'll hear about how to validate protein reagents for DEL, the important characteristics and requirements that are there to ensure that you conduct a screen with meaningful proteins that can that are competent to bind and conduct DEL discovery. I will hear from Svetlana about how to apply, how to use these proteins and design effective selection campaigns. And third, from Letian tomorrow morning, about how to take the output from a DEL selection campaign and how you translate that into DNA, or how you work with those DNA codes and apply the next generation sequencing technology effectively to deliver a DEL screening campaign. Then tomorrow, we'll have a session on data analysis. Once you've made the data, what do you do with this? It's an incredibly cumbersome and challenging situation to be in to analyze one of these data sets. And we'll hear 3 great talks from Paul, Justin, and Gang about how to adapt that sequencing data, turn it into chemoinformatic information, how to leverage that chemoinformatic information to pick the hits out of all the signals that you see, and then finally how to apply concepts in medicinal chemistry to optimize these hits into leads that can be further advanced into drug development. And finally, as I mentioned, we'll have a closing session that discusses emerging trends and technological advances in the field. So operations, how is this going to work? We're operating as a Zoom webinar. Unfortunately, under that paradigm, you attendees will not be able to unmute. So please post all questions that you have for the speakers and panelists in the Q&A box. Now, the session chair will moderate the Q&A at the end of each talk. So the presenter will conclude their talk and then field the questions that have been presented. And you may find that the chairs and presenters are available for one-on-one discussions through using the chat box directly. So I want to say very quickly, thank you to all of our speakers and their home organizations for supporting their participation in this event. And also thank you to those of you who have participants. Wanted to tell you a few things about yourself. With more than 540 registrants, and the last I checked, over 300 people actively signed in participating in the workshop at this point. You guys cover 4 out of the 7 continents on the globe, and more than at least 8 global time zones. Now, breakup here of your where you all come from. Got about 25% of you are academic and government researchers, and the other 75% are big pharma and biotech and chemical industry colleagues. Now, there is also a non-zero portion of you out there who are consultants and venture capital and investment firm colleagues. So I welcome you all to this event. I hope you develop a greater appreciation for the field and can understand more about it. So without that, I will say thank you again to Serine for the invitation to develop a workshop here for you all, and I hope you all enjoy.
Thank you so much, Tim, for introducing the workshop. We have a few minutes and we have a few questions actually, so I'm going to run some of these questions. So the first one is, can target molecules be used in native conformations? Example, receptors in lipid membrane. Do you want to comment on that briefly?
I'll comment to say that I think Svetlana's talk will cover that in detail. So stay tuned.
Okay, and the next question I'll take, it says, would there be a recording available after the workshop? And the answer is yes. This workshop is being recorded and we will have this available for you all on our website. We'll email all attendees and we'll let you know where what site to reach the recordings at. Okay, thank you again, Tim.